In this video, I'm going to cover the histology of the heart. So we'll talk about heart cells, which are called cardiomyocytes. We'll look at them under a microscope. We'll talk about their defining features and what those features are for. And I'm going to start with a diagram because the cartoon diagrams are simpler. They're easier to look at than true cardiomyocytes. So we'll start here. So you can see these are branching cells. They're short, they're thick, and they're branching. And that branching nature is really important because it allows each cardiomyocyte to keep contact, to form contacts with many other cardiomyocytes. Now the heart functions as a single unit, and the cardiomyocytes, their ability to cooperate with all the other cells in the heart is enhanced by their ability to branch and make contact with more than one other cell, with many other cells. So each cardiomyocyte contacts several others, forming a network throughout the heart chamber pairs. So the two atria, they are composed of one network of branching cardiomyocytes, and the two ventricles, they are composed of another another network of branching cardiomyocytes. Most cardiomyocytes have only one nucleus, and that's shown by these actually not that one, by some of these here. Humans have mostly single-celled cardiomyocytes, but about 1/3 of our heart cells have more than one nucleus. So you can see that here. This is actually, one cell. You can see this is all one cell. And so they can have 1/3 of our heart cells have 2 or more nuclei. So the branching structure is visible under a microscope, so that's a defining feature. And most of the ones you see will be single nuclei, but you might see more than one. That's still okay for the heart. Another thing you'll notice under a microscope is these striations, and you can kind of see them here. So this is striated, these cardiomyocytes. And so that striation is also visible under a microscope. That is also a defining feature. And that has to do with the sarcomeres. So if you can remember from Anatomy and Physiology 1, how skeletal muscle works, skeletal muscle also has these striations for the muscle contractions from those through those sarcomeres. The sarcomeres look striped. So here you see a microscopy image on the right, rendered into a cartoon diagram on the left. So I like these kinds of pairs because they really help us understand how what we see in our textbook, which is mostly diagrams, how they relate to what you would see in the real world under a microscope. So again, I'm going to point out the defining features. So you can see these striations. They're not as distinct as you would see in skeletal muscle, but they are there. So you can see these light and dark striations. So striations, I'll just write these defining features up here. Striations. Defining features. And those are those muscle fibers, the sarcomeres that help them contract so that the heart can beat for you. Now, like I said in the last slide, sometimes you will find double nuclei, but you can see here that all of these have single nuclei. And sometimes, like staining's not perfect, so that's why I said, look at this drawn diagram. Every single cell you can see the nucleus, but if you look on the microscopy image, it's not so easy to find all the nuclei. So this is why it's important to look at microscopy images for studying, because it does, things in real life don't look as perfectly as they look in an artist's drawing, because the artists are trying to bring out the most important features. And that's not necessarily how things really look. It's not always as easy as that, right? So usually one nucleus, though sometimes could be 2. One nuclei, usually. Branching. I would say that the striations, the light striations, and the branching are the two of the most important features to help you identify under a microscope. So you can really see this branching nature. It's very different than skeletal muscle, right? But then one last thing are these intercalated discs. There it is there. The intercalated discs are special junctions between cardiomyocytes. And they have a lot of important functions for these cells because the heart, there's a wave of contractions that moves through the heart that helps the blood move in the correct direction. First, you have contraction at the top in the atria, and the contraction moves down through the heart to the ventricles in this wave-like motion and out again, to move the blood out again. And the reason the heart has this coordinated movement is because of these intercalated discs and the special features between these heart cells, between these cardiomyocytes, that allows the cardiomyocytes to really communicate well with one another. Now, the heart doesn't rely on our nervous system to beat. Now, if you're talking about skeletal muscle, every skeletal muscle cell has to be innervated by a nerve in order for it to contract. This is not true for the heart. The heart creates its own action potentials. So we say that the heart is autorhythmic. So if you pretended like you were in that Netflix series, Once Upon a Time, and you could just like plunge your hand into somebody's chest and rip out their heart, it would keep beating. It can keep, those muscles in the heart can keep contracting without the help of our nervous system. They create their own action potentials that drive its own contraction. So it's a little bit like, the heart doesn't have a mind of its own, but it has its own electricity that drives its movement. Now, the thing that will make the heart stop beating is a loss of oxygen, because once it's disconnected from your circulatory system, from your vascular system, it's no longer getting blood, it's no longer getting new oxygen. But it creates its own action potentials, it can beat on its own. It is connected to our nervous system, and what our nervous system does, our autonomic nervous system, our parasympathetic and our sympathetic nervous system, can decrease or increase our heart rate. So our autonomic nervous system can modify our heart rate, but it's not necessary for our heart to beat at a normal base rate. So we say that the heart is autorhythmic. And part of the reason the heart is autorhythmic is because of these intercalated discs. And as you can see here, you can see them under a microscope. There are these strong divisions between these branching cells. That the action potential created in one place in the heart can spread through the intercalated discs to the rest of the heart tissue in a wave-like fashion, creating this wave of contraction that squeezes the blood out through the heart, sort of like you would squeeze you should always squeeze toothpaste from the end. So you have this tube of toothpaste, you start at one end and squeeze it towards the opening of the toothpaste in a wave-like fashion. That's how blood is moved through the heart. So let's talk a little bit more about these intercalated discs. The intercalated discs have three important features. The first is desmosomes. These are a kind of junction between cells, and you very typically find them in other kinds of muscle as well. So they're in cardiac muscle, they're in skeletal muscle. This is how desmosomes work. So you have these two cells, right? Oh, yeah. So here's these two cells. And each cell has a cytoskeleton, which, is responsible in part for maintaining its shape, providing structure to the cell. So these are cytoskeletal elements. So in this drawing, these blue are the cytoskeletal elements. Cytoskeleton. This is the membrane of one cell, like my cell on the right, and this is the membrane of another cell, like my cell on the left. It's the phospholipid bilayer of the cell. Now, the desmosomes are attached to these cytoskeleton elements. And these are desmosomes, there's spellings right there. So you have these large, what's called plaques. So these like pads. The pads are attached to the blue cytoskeletal elements on one side. And then on the other side, there's these proteins, these adhesion proteins between them. So is that labeled? Let's see. Well, they're called connecting filaments here, but you could call them adhesion proteins. Adhesion proteins. So these are not very tight junctions. They don't keep the cells super close together. Things can leak or move between, small things could pass between these two cells. However, they are incredibly strong junctions because it's almost as if like I could reach out to you with my hand and a little anchoring protein or bone would sprout out from my hand bones. They'd be attached to my skeleton inside my body, protrude through my skin, protrude through your skin, and then attach to your skeleton. So like if our two skeletons were joined, we'd be inseparable, right? So that's what these desmosomes are like. They attach the cytoskeleton, which is the cell's skeleton, to the cytoskeleton of the neighboring cell. These are incredibly strong junctions. You find them in places where tissues have to withstand a lot of tearing, right? A lot of torque. So muscle cells, right? So that makes a very, very strong junction. So desmosomes, that's the first thing that you find in these intercalated discs. Let's look at the next thing that you find. Oops. Next thing you find are gap junctions. And this is the most important feature for autorhythmicity or for passing an action potential from one cell to another. Autorhythmicity is set by certain cells, but the action potential created by certain cells is passed to all the other cells, allowing them to work in unison and giving the heart functional syncytium through these gap junctions. So gap junctions are another very important connection between cells found in the intercalated discs of cardiomyocytes. So we have the desmosomes and we have the gap junctions. Gap junctions are pores. They are tunnels that connect the cytoplasm between cells. So it connects cytoplasm, which is just the liquid, right, inside the cells, between cells. So this means that the cells of the heart can share everything, including action potentials. Anything in the cytoplasm of cell 1 can pass to cell 2 and vice versa. So that's shown, these are all different colors because most of the time when we have pores into or out of a cell, the channels are very specific. Like aquaporins only allow water through. You have channels that are only for sugar, only for one kind of ion. Gap junctions are indiscriminate. They just allow sharing of the cytoplasm. The last thing that's important about the intercalated discs, this sort of like moves off, sorry, my image isn't as visible as I'd like it to be because my picture's in the way. But you can still see enough here. So you have the gap junctions down here. That's what we looked at here. You have desmosomes also here. That's what we talked about there, those mechanical connections. So gap junctions allow communication between cells, a sharing of cytoplasm. Desmosomes are a mechanical connection. And then another thing that's an important feature is this waviness. We call that wavy membrane interdigitating folds. Interdigitating folds. How do you remember that? Well, you can see that they're kind of like folds, right? Like wrinkles. Interdigitating, so inter means between, digits are like your fingers. So it's like, if you can imagine this cardiac cell is bumped up against another cardiac cell in this empty space here, and they fit, all these folds fit together. So it's kind of like this. And that's just to increase the surface area of the connection between cells. That increases the number of desmosomes, but most importantly, perhaps, it increases the number of gap junctions, allowing for even more sharing between those cells. So, important qualities of the heart are that it is autorhythmic, meaning that it can create it sets its own rhythm it creates its own action potentials and sets its own pace. Our nervous system does alter the pace. It adjusts it. It can slightly increase or decrease the rhythm of our heartbeat and the frequency of those action potentials that drive muscular contraction. But the base pace is set by the heart itself, and the nervous system is not actually needed to keep the heart beating. Only the vascular system, which supplies the oxygen for energy, is required to keep the heart beating. Also, the heart has functional syncytium. Functional syncytium. It's a strange word. What that means is that all the cells of the heart function together as a single unit. This is due to the intercalated discs. So you have these special pacemaker cells in the heart. They're just modified cardiomyocytes. They are not nerves at all. They're cardiomyocytes that have been modified for creating action potentials instead of for contraction. So the special pacemaker cardiomyocytes create an action potential, but those intercalated discs are really necessary to spread. Once the action potential is created, it needs to be spread through all the tissues of the heart. So this is created by the intercalated discs. And so these intercalated discs spread action potentials, which I'll designate by AP, through the heart, giving it functional syncytium, which means all the cells work in unison. So the pacemaker cells create action potentials. APs. So the pacemaker cells create action potentials, and the intercalated discs allow those action potentials to spread through the myocardium, or the heart muscle. So let's move to the next slide. Here's more microscopy images. Again, these are just from Google image searches for cardiomyocytes microscopy or cardiomyocytes histology. Now you can see again these nuclei. These are other ways these muscle cells can look. So you can see intercalated discs here. You can see this branching pattern, right, of these cells. And one thing I want to point out that we didn't really see in the other images is that the nuclei are often surrounded in white. Surrounded by white. Not always. That wasn't really apparent on the previous histology slides that I showed, but you can really see them, this white here. And what that white is glycogen. Glycogen is a carbohydrate, kind of like starch, but found in animals. It's long chains of glucose. This is an energy source. This energy is to drive the mitochondria. So remember, we eat food, strip it of high energy electrons, which drive the high energy electrons drive ATP production in the electron transport chain in the mitochondria. So we need that ATP production to energize the cells. Now your heart especially, because your heart's been beating before you were born, and it never gets a rest. It's going to keep beating until you die. So it's not allowed to rest, even when it's tired. It can't get tired. It can't get tired. So it may not surprise you to learn that cardiomyocytes have far more mitochondria than other cells. So so cardiomyocytes, these muscle cells, the heart muscle cells, 25% of their volume is mitochondria. So a quarter of the cell is filled with mitochondria. And to give you a frame of reference, when we're talking about skeletal muscle, it's only 2% mitochondria. The glycogen is an energy source for the mitochondria So the mitochondria take the energy from the glycogen and put it into ATP, which is the energy currency of every cell, as you know. So glycogen has the high energy electrons. Those high energy electrons are used to drive the electron transport chain in the mitochondria to generate ATP so that the heart does not get tired. And so just to look at the second picture here, you can see again, this cell's highlighted and labeled for you. So you can see those striations, you can see the white glycogen around the nucleus, you can see the intercalated discs around all the borders that attach this cell to all the others. You can see that branching structure even though it's not labeled, but it's branched, not like long branches, but sort of like the edges of a log. So it's sort of like has these truncated offshoots, these very truncated branches, like a dead log. Sometimes, depending upon stain, what stain is used or how well the stain took, the intercalated discs can be hard to see. So here's an example of that. If you were to look at this whole slide here, it might be a little hard to tell that it's cardiac muscle if you're looking for intercalated discs. Now, if you really zoom in, like on this square, you can see it here. You can kind of see it here, but it's kind of faint. It's hard to find them in some. So if you got a slide like this, you'd have to interpret what it is by this branching structure. You could still see the striations from the sarcomeres. This is why it's good to look at a lot of images. You have to gain an intuition for how these look under a microscope. And look at all different parts of the slide. Here the striations are really clear. Right? You can really see them in some parts of the slide. They're kind of harder to see in others. And that's heart histology, cardiac histology.